Building on the same bowl-focused ultrasonication core, the BoFU family covers different throughput needs and application scenarios. BoFU 1600 is designed for high-throughput sequencing labs that need reproducible fragmentation across large batches. BoFU 100 is a compact single-sample instrument for low-throughput labs that still require precise, highly consistent fragmentation for genetic research and diagnostic workflows. BoFU 80 brings focused ultrasonication into microbial and pathogen applications, for example microbial genomics workflows, and MALDI-TOF upstream sample preparation, helping labs obtain cleaner spectra and more reliable, mass-spec-compatible sample processing. Together, these three systems offer a unified and scalable solution for DNA fragmentation across a wide range of research and diagnostic settings. All BoFU instruments share the same bowl-focused ultrasonication technology, so the differences are really about throughput and cooling design, which map directly to different customer types. BoFU 100 uses built-in semiconductor cooling from 4 to 25 degrees C and handles one tube at a time. It's compact and quiet, ideal for genetic labs or diagnostics labs that mainly do single-sample or low-throughput work. BoFU 1600 keeps the same acoustic power but connects to an external recirculating chiller, also 4 to 25 degrees C, and processes 1 to 16 tubes in parallel. This is the natural fit for core labs and sequencing centers that your MGI teams already work with. BoFU 80 also uses an external bath but is tuned for single-sample microbial and pathogen processing, for example upstream of MALDI-TOF or MNGS. All three are 100W systems with a full touchscreen interface. They run as standalone instruments without a control PC or extra software. For your distributors and end users, that means simpler installation, fewer IT issues, and one learning curve across the whole BoFU family. Here you can see how BoFU actually sits in real laboratories. BoFU 100 occupies about the same footprint as a small benchtop analyzer. Cooling and control are all inside the unit, so customers just plug in power and start working. This is attractive for MGI customers who may not have spare bench space or dedicated chillers. BoFU 1600 offers a bigger capacity but still keeps a compact layout, the main unit, plus a small external chiller. In many labs, it sits side by side with a Cavaris M220. And the overall footprint is very similar. What you can emphasize in conversations is that customers get up to 16 tube throughput in almost the same space where they used to run single tube fragmentation. So for your sales discussions, an easy message is, if the lab has space for an M220, they already have space for a BoFU 1600 and higher throughput. BoFU 1600 was designed to be easy for technicians, not just for PhDs. Everything is controlled from the built-in touchscreen, no external PC, no extra cables. Users have two simple modes. They can set independent parameters for each of the 16 lanes when they want to fine-tune fragment size for different projects, or they can apply one program to all lanes when they are running routine batches for the same panel, so the lab doesn't have to choose between flexibility and standardization. You can position BoFU 1600 to core labs and reference centers as one instrument that covers both method development and high-throughput routine work. BoFU 1600 is delivered as a complete system, not just a main unit. The tube holder aligns each sample at the correct focal point, and the instrument drives the tubes through the acoustic field in a controlled sequence, which is how we keep fragmentation so consistent. Standard accessories, tube racks, centrifuge adapters, cooling connectors, calibration tools are all included. That means you don't need to customize hardware or source extra parts when you install it. In most cases, you can set up the system, train the lab in one session, and they're ready to start fragmenting DNA for their MGI workflows. BoFU 100 is the entry point into focused ultrasonication for many labs. It targets customers who mainly run one sample at a time, hospital labs, small oncology groups, or new NGS labs working with MGI instruments. Cooling is handled by an integrated water bath with semiconductor control from 4 to 25 degrees C, so temperature stays stable even during repeated runs. The dedicated BoFU tubes, Fiber 50 and Fiber 100, are leak-free and easy to handle, which reduces contamination risk and gives good reproducibility even for less experienced users. When you position the product, you can think of BoFU 100 as Cavaris-class performance in a single sample box that fits easily into a clinician's lab or a small research group. BoFU 80 focuses on microbiology and infectious disease workflows. It runs preset, contact-free, temperature-controlled programs that take roughly 3 to 5 minutes during the whole process, with one-button operation. Because the colony sample stays in a closed tube and no probe ever touches the liquid, there is no risk of probe carryover between samples and far fewer tube openings, which makes the workflow safer and much easier for routine lab technicians. BoFU 80 uses dedicated BoFU tube beads, which are pre-filled with glass beads for efficient cell disruption. With these tubes, the system can reliably lyse gram-positive and gram-negative bacteria, fungi, yeasts, and mycobacteria. The spectral results match Bruker's recommended MALDI-TOF protocols, so laboratories can replace a long, multi-step manual workflow with a much faster BoFU 80 step and still trust the ID result. For MGI teams working with hospitals or reference labs that have both MALDI-TOF and NGS, this instrument can be positioned as a shared front-end platform for MALDI-TOF, qPCR, and MNGS. Putting the portfolio together, the key value of BoFU is stable physics instead of fragile chemistry. All models generate a uniform cavitation field that can be tuned from about 100 BP up to 3000 BP, giving labs much more control over insert size than typical enzymatic kits. Because fragmentation is a physical process, the results are independent of reagent chemistry or chemical conditions, incubation timing, or who happens to be on shift that day. Typical DNA recovery is 60–95%, which is especially important for oncology samples, FFPE blocks, and any low-input project on MGI platforms. And regardless of whether the user is running a single tube or a 16-sample batch, the operation stays the same. Choose a program on the touchscreen and press Start. This is exactly the opposite of what many labs experience with enzymatic fragmentation, and it sets the stage for why they are actively looking for a more stable way to fragment DNA.